And while you're doing that, I'll go ahead and introduce you. So Mackenzie Gunn, she received her undergraduate degree at the University of New Hampshire in marine biology and food sustainability. And she's working on her master's right now at Mississippi State in wildlife fisheries and aquaculture, specifically on the catfish trematode infection in farm-raised catfish, under the direction of Peter Allen and Matt Griffin. So take it away, Mackenzie.
Thank you. I actually just completed my master's in May, sort of a weird ending to it, but I'm officially done, so that's kind of nice. So this presentation is a piece of my master's thesis looking at the comparative mortality and parasite-induced anemia in channel and hybrid catfish exposed to Bulbophoris damnificus cercaria. Can't change my slide. Sorry, I don't know what is going on. Okay, so just as a little bit of a background about catfish farming in general, catfish farming was established in the 1940s, primarily for stocking into lakes, rivers, and reservoirs. By the mid-1960s, channel catfish took over as a predominant food fish in the United States. And today, catfish farming is the most economically valuable aquaculture market in the US, with producers grossing over $360 million in 2018. The majority of catfish farms are located in the southeastern United States, primarily in Mississippi, Alabama, Arkansas, and Louisiana. And as catfish farming has intensified in past years, hybrid catfish have started to become more popular for producers to be using. The hybrid catfish is a cross between a channel catfish male and a female and a blue catfish male. The channel catfish was produced to be selected for production optimization. This includes higher tolerance to low dissolved oxygen, higher tolerance to crowding, a higher feed conversion ratio, and most importantly for this talk is that hybrid catfish tend to be resistant to some pretty common channel catfish diseases. However, there are some diseases that haven't yet been studied in hybrid catfish, and one of those is Bulbophoris damnificus that I'll be talking about today. So Bulbophoris damnificus is a trematode parasite that was first identified in catfish ponds in the 1990s. There's actually two species of Bulbophoris damnificus, or of Bulbophoris damnificus and Bulbophoris species type 2. Bulbophoris species type 2, though, has not been shown to infect channel catfish, and so it's not typically a species of concern within catfish ponds and for producers. So for today's talk, if I say Bulbophoris, I'm talking about Bulbophoris damnificus. The life cycle of Bulbophoris damnificus starts with the adult living in the digestive tract of the American white pelican. When the pelican defecates in the pond, it releases eggs. These eggs then hatch as miracidia, which infect the ram's horn snail and mature into cercaria. The cercaria are then released into the pond and infect the catfish within the pond. These encyst and mature into metacercaria, and the life cycle is complete when a pelican eats an infected catfish. Bulbophoris damnificus has been shown to have pretty high economic impact on producers, and ponds, even with a low to moderate infection, can have up to 61% in economic loss. Catfish that are infected with Bulbophoris damnificus typically present with distended abdomens, hemorrhage, exophthalmia, and disease-induced inappetence. There's a parasitic encapsulation period from 7 to 14 days post-infection that's been seen in channel catfish, and a peak mortality between 9 to 15 days post-infection. However, when fish are removed from a source of new infection, recovery has been shown to be possible, and growth of infected channel catfish was shown to be similar and sometimes better than unexposed catfish. There's no treatment for Bulbophoris damnificus infection. Once the catfish are infected, they just are infected. However, a way to mitigate disease is to prevent further infection. This is typically done using a chemical treatment of the pond, such as copper or hydrated lime slurry. This is done to kill the snails, which breaks the life cycle of the parasite and prevents further infection of the fish. However, these chemical treatments are typically pretty expensive for the farmer and also very stressful for the fish. And so producers usually only will treat ponds when a heavy infection is observed in the fish. However, there's a new problem that's been arising for producers of hybrid catfish specifically. This is a catfish that was brought to the processor and was turned away, obviously. The fish was absolutely riddled with trematodes. However, the producer didn't know that they had a problem. So hybrid catfish producers are now realizing that hybrids may not have the same reaction to bulbophoros infection that channel catfish do. And so the typical signs that a producer would look for in channel catfish might be missed in hybrid catfish. And so there's large economic losses that are happening, but it's at the end of the production cycle and the producer doesn't have the chance to treat snails and potentially lessen the impacts of this trematode. A study done during my master's showed that viable metacercaria were present in hybrid catfish for up to 13 months. We ended our study after that, so it could potentially be longer, but the typical production cycle is 18 to 24 months, meaning that a catfish that's infected early on in the production cycle can hold that infection throughout the entire production cycle. Because we're not sure what the difference is or why hybrids might have a lesser impact from bulbophoros, we wanted to know what the actual differences were between channel and hybrid catfish. So the objectives of the study were to assess the difference in mortality between infected channel and hybrid catfish, as well as to try and understand the difference in physiological response between channel and hybrid catfish. So for both studies, the first thing we needed to do was collect cercaria. Over the course of my master's, which played into these two studies, 10,473 snails were collected from ponds throughout the Mississippi Delta. Each snail was held individually for 48 hours in a vial and was visually checked for cercaria every 12 to 24 hours for about 2 to 3 days. Any snails that were shedding cercaria that were visually confirmed to be a bulbophoros-like cercaria were molecularly confirmed using a duplex qPCR as either Bulbophoros damnificus or Bulbophoros species type 2. So for the mortality challenge, we actually did 2 challenges. Both had the same experimental design. There were 5 exposed tanks and 1 control tank per species. For the first challenge, we used smaller fish, 0.3 to 7.1 grams. These were stocked at 30 fish per aquaria and challenged with 100 cercaria per liter. They were observed for 15 days. For the second challenge, we used slightly larger fish, 5 to 23 grams. These were stocked at 20 fish per aquaria and challenged with 250 cercaria per liter and had a 60-day observation period. I will note that any fish that were moribund but not actually dead in the tank were left because there is the possibility of recovery after infection, and so we wanted to make sure that any fish that had the chance to survive were able to. So this is the results from that study, from both of those studies. The period in gold is that 7 to 14 days post-infection encapsulation period. Both studies had a similar trend where hybrid catfish had significantly lower mortality overall. As you can see, there's a pretty significant peak and then a leveling off of mortality during and then slightly after the encapsulation period. And the peak mortality in all challenges was during that 7 to 14 days post-infection. In channel catfish, it was 6 days and 10 days post-infection, and in hybrid catfish, it was at 8 days post-infection. However, in the second challenge with the larger fish, there was a steady loss of 1 to 4 fish per day on days 1 through 25 post-challenge, and that prolonged chronic mortality that was observed is not typically found from bulbophoros infection. So that was just sort of an interesting finding in that study. Overall, the cause of mortality from infection is likely due to the osmotic and homeostatic disruption, but the physiological response is not known, and the physiological reason that hybrids may have a lower mortality is also not known. which leads to our second objective, to understand the difference in physiological response between channel and hybrid catfish. So in parasitic infections or trematode infections, there's a neovascularization that's associated with the encapsulation process. This isn't unique to Bulbophorus, but it is thought to transport nutrients and waste to and from the parasite and host. There's anecdotal evidence that's been seen for an anemic response to Bulbophorus damnificus infection in channel catfish, but nothing had been studied up until this study. The experimental design for this was the setup here. There was 3 control tanks and 4 exposed tanks. Out of the exposed tanks, 3 were sampling and 1 was observation. This tank was not sampled out of, it was just used to estimate mortality with the same cercaria concentration. These fish were observed for 48 days post-infection. They were sampled during 9 different days over that period. On each sampling day, 2 fish per tank were sampled. They were euthanized with MS222, a weight and length were taken, and they were bled through the caudal vein. The parameters that we looked at were whole blood, hematocrit, and hemoglobin, and in the plasma were osmolality, glucose, and lactate. So for the whole blood parameters, this gold bar again is the 7 to 14 days post-infection, showing that encapsulation period. There was no significant difference between channel and hybrid catfish in terms of physiological response in any of the parameters. So although these figures are broken out into channel and hybrid catfish, the significance is actually just between control and exposed treatments. So for hematocrit and hemoglobin, there was a significant difference between control and exposed treatments on days 11, 14, and 21 post-infection, with a pretty drastic drop in both hematocrit and hemoglobin, and then a subsequent recovery that can be seen. Red blood cell concentration followed a similar trend with significant decrease in exposed fish concentration at 11 to 14 days post-exposure, which is again during that encapsulation period. There was no significant differences found by day in any of the plasma parameters. But overall, there was an anemic response that was seen during the encapsulation period, as well as a subsequent recovery. This is sort of just an interesting, these photos on the left here is the gill and blood from a unexposed catfish, and on the right is, I think around day 11 from an exposed catfish. As you can see, the gill and the blood are both very pale and it was a pretty drastic anemia that was seen. Overall, there was no mortality in any of the control tanks, and there was a higher survival of hybrid catfish over time. I think out of our sampling tanks, there was only 2 or 3 hybrid catfish that died during the course of the experiment versus between 7 and 10 channel catfish. Overall, there was no change over time in glucose, lactate, and osmolality. However, glucose was lower overall in both channel catfish and exposed treatments just over the course of the whole study. This could be due to behavioral difference or disease-induced inappetence. Like I mentioned before, that's a typical clinical sign of trematode infection. However, during our study, we noticed that the hybrid catfish really didn't go off of feed at all. They also were very active in the tank during the entire study, whereas during peak mortality, the channel catfish really didn't eat and hung pretty low to the bottom of the tank. There was an overall decrease in exposed fish osmolality. This could be indicative of a long-term low-level stress response to the parasite versus an acute stress response that we may have expected to see from infection. However, we had a pretty small sample size of only 2 fish per tank on the sampling days, and so there's a high degree of individual variability, and so some of the data could be a little bit confusing due to that. Overall, all parasites use host-derived resources. That's what a parasite does. However, there is evidence found in other papers that parasite infection may not generate the same classic stress response that one would expect to see. That's because suppressed immune response could reduce the ability of the fish to respond to the infection. So it is possible that there is a difference in physiological response, just potentially not in the parameters that we were able to look at. Overall, there was a lower mortality in hybrid catfish than channel catfish in both size classes of fish, and no difference found in physiological response due to infection between the two fish. So the disease management implications of this are that farmers, especially with hybrid catfish, need to treat their snails. Treating snails proactively, especially in ponds with known pelican activity, could potentially eliminate the problem of Bulbophoros damnificus infection in hybrid catfish. The other option is to do routine pondside checks, so actually visually observing fish to look for the metacercarial bumps beneath the skin to stay ahead of potential infections within a pond. Infections in hybrids might not be noticed until processing, and so production losses are higher at the end of the production cycle versus being able to mitigate issues in the middle of it. I'd like to give a special thank you to Brian Ott for all of his help during this study, as well as everyone from Mississippi State that's helped, and the NWAC faculty and staff, as well as my funding sources. And that is all. Any questions?
All right, thank you very much, Mackenzie. If folks have questions, go ahead and unmute yourself and ask them, or you can put them in the chat box and I can read them to Mackenzie.
Hey, this is Jan from Fish and Wildlife in New Jersey. How you doing? That was a nice presentation, Mackenzie. Thank you. One question about blue catfish. Do you know how susceptible blue catfish are to Bulbophoros?
Blue catfish, so Dr. Griffin did a study looking at channels, hybrids, and blue catfish, and they were all equally susceptible to Bulbophoros infection. But in terms of mortality, I'm not sure like what the impact is that in that sense.
Okay, so they're not resistant to Bulbophoros, though they're just as susceptible.
Yeah.
And then a second question, just relating to aquaculture management strategies. Do pond owners do any indirect treatments for aquatic vegetation to control snails, or is it always chemical treatments?
So I know that like keeping the edges of the pond mowed do reduce habitat for snails, like up on the edges. But one of the problems with that also is if there's snails in the pond, that they can congregate towards the middle. So it might just be that you're creating an environment where you're not seeing them versus them actually not being there. Does that answer that question?
Yeah.
Okay.
Great, thank you.
Mm-hmm.
Hi, Mackenzie. This is Gail Kirath out in Seattle. Again, another really nice talk. Thank you. I was particularly interested in the difference in mortality that you saw in your challenge 2 with the bigger fish. It seems like there's definitely a sort of an age-associated difference. So it still looks like there's a big difference between the hybrids and the channels as long as they're above a certain size, right? So I had 2 questions related to that. You showed that one ugly fish that the producer had taken to the processor but didn't realize it. So does that suggest that there's quite a bit of, do you think that the lower mortality you saw in challenge 2 reflects what's happening out there a lot, and so there might be more infection cycles going on, but people aren't seeing them because it's not killing the fish?
Yeah, it absolutely could be. Typically, losses from Bulbophoros are seen in fingerlings and smaller fish. I think part of that is just body size. I mean, if you have the same infection to 2 fish that are, one's very small and one's a lot larger, the smaller fish is definitely going to be more impacted by that. So it is possible that there are ponds with moderate to low infections, especially in hybrids, where you wouldn't necessarily see any impact from that until you actually pull the fish out and look at it.
So does that, my second thought then is, does that suggest that the advice from managing might be to make sure you do the snail control as juveniles, right? I mean, there's an age class distinction in the need for snail control, maybe.
Yeah, absolutely. I think, but even in ponds that have larger fish or food-sized fish can have snails that are shedding cercaria, and so you could potentially still have the same problem with ending up with fish that are just riddled with trematodes, which are, so even if you're not getting the losses due to mortality like in the smaller fish, you're still getting losses because you've fed these fish all the way through the production cycle, and they show up to the processor and you can't sell them.
Right.
Yeah.
Good thoughts. Thanks.
Yeah.
All right, are there any other questions for Mackenzie?
Surely Dr. Soto has a question.
We don't have enough time. There's only 10 minutes.
Very funny. Very funny. Great job, Mackenzie.
Thank you.
I don't have a question.
Excellent talk. I can ask one, Matt and Esteban. This is Haitham from University of Wisconsin. Do you know if there is any effect for temperature on the infection rate, and what temperature you used for your challenges?
The temperature for our challenges, I cannot remember off the top of my head. It was, I want to say around 26. I'm not sure though. I would have to look, but it's not necessarily infectivity that changes with temperature, but the snails start shedding in the spring as the temperatures warm up, and so it is possible that there is some effect due to temperature.
Okay. Thank you.
Yep.
All right, well, thank you very much, Mackenzie. We'll go ahead
Thanks.
and wrap it up.